first speaker, Dr. Juergen Richt. Dr. Juergen is a Regents and University Distinguished Professor at Kansas State University since 2008. In 2010, he became the director of the Department of Homeland Security Center for Excellence in Emerging and Zoonotic Animal Diseases, and in 2020 was the director of the National Institutes of Health Center for Emerging and Zoonotic Infectious Diseases. He has edited several books, obtained several patents, and published more than 360 peer-reviewed articles, and raised more than $70 million in grants for veterinary research. Dr. Richt is a pioneer in veterinary science, most notably in the One Health field, and has worked on high consequent pathogens with zoonotic and transboundary potential, leading strategies to identify, control, and eradicate such agents. His basic and applied research includes studies on animal influenza viruses, animal prion diseases, including BSE, Rift Valley fever, African swine fever, monkeypox, and SARS-CoV-2, as well as Borna disease virus. And today he will be presenting on emerging viral threats and mitigation strategies in the swine industry. Thank you for being here, Dr. Richt.
Okay, good morning. Hope everybody had a good evening last night and a good morning. Thank you, Kim, for the introduction and also for the organizing committee, especially Monse, to invite me here. It's my first Leman conference, and we'll see how the next 45 minutes go. Maybe I get invited again. So anyhow, I was asked to talk about viral threats for the swine industry. And my lab is very diverse. We work not only with swine, where are the swine? Here, we work with a lot of different animal species, even wildlife, and with a lot of different viral host models, and many of them are obviously we do in swine. We did SARS, highly pathogenic avian flu. That's my running horse, swine flu, swine influenza. We just finished a study with Japanese encephalitis genotype 4 in pregnant sows last week. We worked with VSV and African swine fever, CSF, and classical swine fever, and even with monkeypox in swine. And some of these diseases I will cover today. So the outline of my presentation is I give you an overview of my limited view on viral diseases for swine of concern, and then we go in detail about how to mitigate them. And I go through a bunch of different mitigation strategies, biosecurities, diagnostic, etc. I get to that in a minute. So just to set the stage, and I think everybody knows that, but I want to remind everybody how important swine viral diseases, they threaten the swine health, productivity, and global trade. And also some of them pose zoonotic risks to public health. And so they are very challenging to the industry for animal health, economy, trade, food security, zoonotic risk I just mentioned. And virus specific is viruses have a lot of opportunities and propensity for evolution. RNA viruses don't have a proofreading mechanism, etc. So evolution is very critical, and you hear about that when we talk about PRRS. PRRS is running away. You have a vaccine or something, and 2 weeks later, the virus is already different. And in some of these diseases we discussed today, there are no vaccines. And I tell you what the reason is for that. And I believe, and I talk about rapid diagnostics, point-of-care diagnostics, I'm really talking about point-of-care diagnostics for a long time, for more than 10 years, how important it is, especially about transboundary diseases, like foot and mouth disease, if it breaks out at a place very far from Plum Island or soon from Manhattan, Kansas. If you can diagnose it at the spot, at least you can stop movement, and you can wait. If you stop movement, the virus doesn't spread and the animals are not distributed as fast. And so I believe, and I come at the end, I will talk about how important integrated strategies are. And I am a scientist, a veterinary scientist, and I have to be part of that goal to develop essential and important strategies to protect the global swine industry. So anyhow, this is just an overview, and you have heard about this a lot, of the transboundary viral diseases. I first or show you some notifiable. Everybody heard about African swine fever. It's knocking on the door. We heard about classical swine fever. We never had that African swine, and we don't have classical swine since 50 years. But we have it in South America, and African swine in the Hispaniola island. Foot and mouth disease never goes away. And we were lucky we didn't have that for the last 100 years. And then these vesicular diseases, vesicular stomatitis, notifiable, vesicular exanthema of swine, swine vesicular disease, they're all notifiable, but some not listed with a WOA. Japanese encephalitis, I just discussed that. It's notifiable, never been reported in the US, but it's mosquito-borne, it's arthropod-borne. So you never know when these arthropod-borne pathogens will hit the US. Menangle in the Pacific Rim, there's just a couple. Nipah, never had here. It's a very dangerous zoonotic pathogen. We had H5N1 even in pigs. We had one case in a backyard farm in Oregon. Rest Ebola in Philippines, never reported here. And something I thought was very interesting, blue eye disease, very prevalent in Mexico, like screwworm. And we all talk about screwworm. When does this one hit us? And then we have a whole slew of endemic viral diseases. And the biggest one you discuss, I think, for hours is porcine reproductive respiratory disease syndrome, PRRS, and half of our herds, this is the what is it? Morrison Swine Health from earlier this year, 50% about of our herd are seropositive either through vaccination or through new infections. And the other one, important one, is swine flu. About 25% of our swine herds are positive, and I will mention that during this talk a couple times, these two diseases. And then we have a whole list of other diseases. Everybody, especially when you're a scientist, you cannot do everything. You have your specific pathogen you want to work on. And so I just listed that here. There are many, many others. And we had a big awakening several years ago with porcine epidemic diarrhea virus. You still might remember that, and the association with feet and other ways for mites to transmit this virus. So anyhow, what are the strategies to prevent, control, and eradicate or eliminate diseases? Number one, biosecurity. And I also want to go into biocontainment because people don't talk so much about biocontainment, but biosecurity is critical. And this has been preached, I would say, for the last couple of years to the swine industry with ASF being on the horizon. Diagnostics, and here I will give examples. I use examples from my own lab and from the literature with different animal diseases which might be of interest to the audience here. Here I talk about point-of-care diagnostic for African swine fever. Vaccines. So I will, obviously they're very critical, and I talk about modified live virus and subunit vaccines, and here examples are African swine fever and swine influenza. And then we talk even about antivirals. There are some diseases we have no vaccines. Maybe antivirals is a solution, especially when they are cheap and effective. And a pig which would die, and you can treat it for $10, $20, and it survives. That's really an economical Gesundheit solution. And then at the end, and I thought I was here yesterday afternoon, we talk about gene editing. And many of you know about these examples, PRRS with the knockout of the receptor. And I give you an example from my own work, my own lab's work with swine influenza to make resilient pigs against diseases. So we will talk about PRRS and swine flu. And hopefully one day we have a pig which is resilient to African swine fever. But that's still far away. So biosecurity and biocontainment, core defense against disease introduction and spread, and critical for all farms regardless of scale. So when we think about biosecurity, we also have to think about, this is not only on the farm, this is on the border. So you need, the beagle brigades you see, very critical. One beagle, to train one beagle costs a lot of money and takes a lot of time. And then they can only work, I think, 3 hours at a time, and then they need a break for 2 hours, and they can work 3 hours again. So we need to spend money to have biosecurity at the international borders. We have biosecurity also at the national and state level. You just remember recently the outbreak with H5N1 in cattle. There was no movement of cattle from state to state if you did, after some time, it took a couple months obviously, if you didn't take a test and they were negative, at least in their milk, when they tested milk. So there is also biosecurity measures on the state level, and then obviously biosecurity in the farm, and all this is a repetition, but it's important to prevent disease entry. We have to talk about animals which come onto the farm, we talk about visitors and staff, we talk about vehicles, equipment, and even feed. And also water, we can discuss that later, and wildlife and pests. Many of these animals, these rodents and vermin, they carry some of the pathogens which our animals are susceptible to. And then we need this biocontainment, internal biosecurity. And that means if you have a sick pig, you have to limit the spread. You cannot just let it go and forget about it. So this is important, and some people use the all-in-all-out production. We have to understand sanitation and disinfection. What is the right disinfectant if you have a specific pathogen which infects your pigs? We have to isolate, if possible, sick animals. And think about workflow. If you send your workers first into the sick animal barn and then to the clean young animals, no, that's a no-no. So you have to think through these things to have biocontainment. And also manure waste management. One of these pathogens, they survive for a long time in manure and waste. And also filtration and ventilation. And also, when we think about ASF and other diseases which can hit the United States, we have to think about emergency preparedness. And this is critical for every operation at the farm. So this is a slide I borrowed from a paper here in 2020. I think it's very nice. This was the past, or should have been the past. You should not have a farm where every movement, vehicle, supplies, equipment, visitor, they go into the farm and go out. And even trucks from abattoir or rendering facility go in and out. That should be the past. We should have farm biosecurity like this, where you have a control, entry zone, here's a risk zone, then you have a buffer zone where you more or less, people shower in, they're not allowed to take food into the facilities, etc., and they have clothes specifically for the clean zone facility. So we have restricted movement, we implement quarantine measures, and we create risk buffer and especially a clean zone. And that needs to be told your workers, needs to be told the people you work with, the suppliers, etc. They need to be educated. Okay, now we go to another mitigation strategy, diagnostics. So diagnostics enable rapid detection of pathogens, and also if you detect your pathogen, rapid responses. And they're essential for surveillance and outbreak control. And one advance in the last 10 years, especially also in veterinary medicine, are the point-of-care diagnostics. What does that mean? These are tests performed in the pen, on the farm, in the field, close to the animal. They provide real-time results without the need of centralized labs. You don't have to ship the samples to your local labs or to your, if it's for an animal disease, to Plum Island or soon to Manhattan, Kansas. And it enables rapid decision-making. We are talking minutes to hours. Minutes with a lateral flow test and hours with PCR, many PCR. And this allows the isolation treatment and culling really fast, and improves surveillance and outbreak containment, and obviously reduces economic and trade losses. And it can be directed towards the nucleic acid, to the genome of the virus, or to the antigen for a specific antigen of the virus, and I'll show you an example, or antibodies. And very critical, I always say that, is proper end-user training. So if we talk about transboundary diseases, it has to be in the hands of USDA, the regulatory agency, USDA-APHIS. And it has to be controlled results reporting, and validation is very critical for these tests. And here are already the challenges. Some of them are not validated. The sensitivity, specificity could be less, and I give you examples for that in a minute. And some of them cost more than laboratory diagnostics, and they need regulatory approval. So I talk about now for 3-4 slides about our approaches to point-of-care diagnostic for African swine fever. And what we did already many years ago, we used point-of-care molecular diagnostic with 2 different machineries. The Pocket from GeneReach, which you can carry as a suitcase. You drop it off, you check it in at the airport, and take it wherever you want to take it. It has a PCR machine, has a centrifuge, pipettes, etc. So you can do everything wherever you go. In this case, you need electricity. The PCR machine can run on a battery, car battery. And then in the last couple of years, we used another machine called Franklin from Biomine, and this one is totally independent of electricity. You charge that machine, it can run 2 cycles of PCR with 9 samples. This has 8 samples. And the RNA, DNA isolation cartridges are all, you use a syringe to go through the different steps, and you can do everything. I have done it in the desert, in the Gobi Desert. The next electricity was maybe 4 hours away by car. And then I show you some data on a lateral flow test for antigen detection for ASF. So why do we need these things? I go often to Africa, South Africa, to validate, and other countries to validate my test system. And here is an example where we went to South Africa, and you all know that the warthog is a virus reservoir for ASF. And they are a pest in some of these game parks, and they bring in hunters, professional hunters, and they obviously kill these animals, and we were able to take samples, blood and lymph node samples, and then you can test for the presence of ASF using your molecular tools or other tools you have available in the field, in the South African, in the bush. And this is just a result when we did that in South Africa with our colleagues from Onderstpoort at University of Pretoria. This is the laboratory thermocycler results. We had 132 positive samples and 60 negative samples. And when we tested them with the Pocket, we had 100% correlation with the lab-based thermocycler. So this looks pretty good. The molecular stuff looks pretty good. So I just summarized this. When we use these assays to validate it with different panels, they look pretty good. We did inclusivity tests with 3 different genotypes, 1, 2, and 10. We did the clinical sensitivity and specificity was pretty high. The biomeme showed similar sensitivity and specificity, so we conclude that these point-of-care molecular devices show equivalent sensitivity and specificity to the lab-based PCR machine. So now you don't want to have all the sophisticated stuff, PCR and whatever. You need sometimes easier method to detect the disease. And in this case, my lab works for many years with African swine fever. We made monoclonal antibodies against different antigens, and we worked together with a company in California, SLRC, to develop a pen-site antigen test. And the concept is simple. You have you analyze your sample. We have our monoclonal antibodies here conjugated with gold, and then that flows over here to our test and positive control line, and then it interacts either with my antibodies here, which are here in these test lines, and then you have a readout. And it's very simple. You only need 10 microliter of blood. It takes 15 to 20 minutes. And it only needs a tube. You add water to that tube, no buffer. And then you insert your strip. You wait 15 minutes, and then you have a visual readout like this. So this is a summary. This was published by my group and also in parallel or confirmed by the group of Amygdala in Canada, Canadian Food Inspection Agency, similar results. So when we used this test, we could detect first time the animals were positive by 4 days post-infection. And they needed, and when we had cell culture supernatant, we needed about 10 to the 4 PCID50. Not as sensitive as PCR, that's very clear. Less sensitive than PCR. Had a pretty high specificity, and when we looked at sensitivity, the sensitivity increases with clinical signs. So it was pretty good with mild to severe clinical African swine fever signs. When we used the genotype 2 virus or genotype 1, it was above 90%. And also very good when we had 10 to the 8 copy numbers per mil in the blood. We had pretty nice sensitivity. So it is not as sensitive as PCR, but it can be done in 10, 15 minutes or 20 minutes in the pen and has a pretty good specificity. Okay, now we go on to vaccines. Vaccines are obviously the cornerstone of viral disease prevention and eradication. There are 2 viruses which are eradicated from human or animal kingdom. One is smallpox. There are only some left in certain labs, and the other one is rinderpest. And both were eradicated using modified live virus vaccines. Very efficient, low, there is always side effects. They had side effects, but they were eradicated. And there are traditional next-generation platforms, and we discuss that later, and obviously a variety of viruses are targeted. So this is a beautiful overview of how vaccines work. And this is the example of African swine fever by Urbana and Ferreira. I borrowed this. So vaccines, you can use a vaccine, take a virus and inactivate it. We call them whole inactivated viruses. You can take a virus which is naturally attenuated. You can use a virus which is attenuated in the field. So you get a virus after whatever, a couple of years, it attenuates itself. Or you get a virus which you attenuate in cell culture or in animal. You passage it 100 times in a rabbit or 100 times in cell culture. Or you use modern technologies where you do homologous recombination or reverse genetics, whatever, and do gene deletion. Or nowadays, obviously, we have DNA or even RNA vaccines. And then there are people who use recombinant subunits. They express it in baculovirus, E. coli, yeast, you name it, even in human cells. And then many people, including myself, we use recombinant viral vectors. We deliver them with the antigen of interest to the animal with a virus. So it is a virus which doesn't make the animal sick, and so the antigen of interest is expressed in a normal viral replication cycle, and it's also used as a vaccine vector in many, many cases. So what do we know of African swine fever vaccine? There is no safe and efficacious vaccine commercially available in non-endemic countries. The only country is Vietnam, and we get back to that later. Why? Inactivated virus formulations do not work. Subunit vaccines, in many cases, I have published several subunits, don't work. They even enhance disease. And then others, different antigens, you have 50 to 60, sometimes 100% protection, but very difficult to replicate. And then we have the MLE vaccine. Everybody talks about modified live virus vaccines for ASF, but are they stable? They are not. Do they reverse to virulence? Some people have shown they reverse to virulence. And then even if you have a good one, how do you produce a lot of these modified live virus vaccines if there are no cell systems available? You have to isolate macrophages from pigs from the slaughterhouses to produce your vaccine virus. There is no good cell system right now, which produces high amount of virus that it's feasible really for production. That's what I understand. Why is this? This virus is highly complex. Has between 150 and 180 oberine frame, and we only know in detail the function of maybe 30, 35 of them. So majority, we don't even know what they do. And many do encode factors, viral factors, which shut down the immune system, whether it's the innate immune system or the adaptive immune system. And the virus is smart, like PRRS. It hides in the macrophage. And it travels with the macrophage throughout the whole body and hides. And that's a big issue, and you know that with PRRS. I don't need to preach to the choir. And then we don't even know the mechanisms of protection, the correlates of protection. Do we need antibodies? And if yes, which? Do we need T cell and what specificity? And is innate immune system important? We do not know correlates of protection. If you want to do a rationally designed vaccine, you need to know the correlate of protection. So everything we do is shooting in the dark, and hopefully we hit the target somehow. And even if we get a vaccine, say against genotype 2, and then a genotype 1 comes around, the genotype 2 vaccine might not protect against the genotype 1 and vice versa. And we have 24 genotypes and 8 serotypes. So there is a lot to be done. And you know how difficult it is for flu to make a universal vaccine against influenza. They talk about it for 40 years, and to date, we don't have one. So this is a paper which was published several years ago by the group, USDA group of Manuel Borca and others. And what they did is they made a modified live virus vaccine by deleting one gene called I177L. So this is the wild type. The genome is 170 kilobases, about 190 in size. And they deleted this gene by homologous recombination. And when they inoculated this deleted modified live vaccine, it was attenuated in different doses and could protect against virulent challenge, lethal challenge. And this is the vaccine which was approved in Vietnam as NAVET ASF-VAC in July 23. There was another one which is a different region, MGF, it's a multiple gene family region. They deleted 6 genes, and that was also approved from another company, AVEC ASF Life. And as I understand, I believe this one is tested right now for safety in the Dominican Republic and the Philippines. There's a lot of discussion about this vaccine safety. Some people have shown reversion to virulence. Some people believe they are not safe. But anyhow, time will tell. The most important thing is, about 3 years ago, the Chinese recognized a new recombinant virus. A recombinant between a genotype 1 and a genotype 2 virus. And this virus is already in Vietnam and other countries in Asia. And none of these vaccines which were produced against the genotype 2 nor against the genotype 1 protect. So the virus, a virus which was evolutionary, very silent, there was not much happening, is now getting, it mutates, it recombines, because we try to intervene and do things which the virus doesn't like. So this is the presence of ASF. Evolutionary, really quiet virus is becoming loud and dangerous, and even the vaccines they produce don't work anymore against the new strains. So we work on subunit. I'm not a big, I'm nervous with these modified live virus vaccines in the field, so I decided early on to work on subunit, very tough. Not easy, but we have some light at the end of the tunnel, a publication we have together submitted or in review with Dr. Mwangi's group, where we cloned all 180 open reading frames in 43 adenovirus, so it's a vectored virus constructs. So we have 43 constructs with multi-cistronic open reading frames. And when we use that in different groups, we have used all 43 constructs plus Quill A. We have less constructs, only 40 plus Quill A. Then we used 43 constructs without adjuvants, and then we have a control, the GFP construct with adjuvants. And the results are summarized here. This is our control. We have contact infection. These are the empty vector controls in purple. They get all sick or die within 14 days. The group which had the adjuvant plus all constructs plus the adjuvant also dies within 14 days, and the same is true with the group which had less adenovirus constructs and the adjuvant QuIL-A. But the group with all the constructs but without the adjuvants, 5 out of 6 or 6 out of 7 of these animals survived. So there's the end of a long, long, long tunnel, because we have 43 constructs to go through, and look what is protective immunity. But there is some light at the end of this tunnel. When I came to this country maybe 25 years ago, I started to develop a modified live virus vaccine against flu by using reverse genetics and knocking out or truncating the NS1 protein, an important immunomodulatory protein which interacts with the type 1 interferons. And this works beautiful after IM inoculation and also IN inoculation, even in the presence of maternal antibodies. And Boehringer Ingelheim licensed it and sold it at Ingelvac Provenza for several years. But this was stopped because obviously it is a live virus and has the potential to reassort with field viruses. But there are some solution to that. And my friend or my colleague Daniel Paris just published a paper making a modified live virus vaccines for H9N2 avian influenza virus. And what he did is to make that vaccine more safe by make it reassortant impaired, non-transmissible, by knocking out one open reading frame for the M2, this is a transmembrane protein, and move that open reading frame to a different gene, to the BP1 gene, and adding some sequences into the HA, next to the HA signal peptide. So this virus is has reduced reassortment capabilities. So there are solutions to these problems for the future. And modified live virus vaccines that I told you are very efficient. They are the ones which were used for eradication of several diseases. So now we move on to something I would not have talked about 4 or 5 years ago, antivirals. Do we need antivirals for animal therapy? Do we use them in the animal kingdom? Yes, we do it for some time, especially in pet animals, like Tamiflu with H5N1, etc. But it's something like that, an option for swine. And I say, yes, if we have a disease which we cannot cure and the animal dies, and I have an option to use a drug for $10, $20, and my animal survives, yes, it is an option. So we, even in the swine industry or in the livestock industry, have to think about antiviral options. When I started virology, nobody thought about antiviral therapy. Now, we have antiviral therapy against HIV, we have antiviral therapy against HCV, we have antiviral therapy against flu, against herpes, and so on. So we need to be open and think about it, because if we can reduce viral replication and death, and it's feasible, economically feasible, that's even for the swine industry an option. So I want to give you some examples from the literature. I don't work much on antivirals, where antivirals were used against African swine fever virus. And I show you even antiviral systems. CRISPR-Cas can be used for antiviral therapy. We are working on this too, and I show you some examples how people have used it. And here we talk about a drug I have trouble to pronounce, brinsitofovir. It's a drug used, the brand name is Tembexa, and it's used to treat smallpox or Mpox patients. We had a lot of 40,000 Mpox patients a couple of years ago. So this drug was used to treat them. However, this is the problem. 100 milligrams cost $1,000 when you buy it in the pharmacy, right? But obviously you can produce it cheaper if you get generic drugs. But anyhow, you have to start somewhere, right? And a group in China, they used this to test it against African swine fever. And as you see here, this is very simple. This is the control, and here you use more dosages of this drug. You have less replication, and also, I don't know whether you see it here, less virus, they don't light up so much if you have a higher dose. But most importantly, what they did is they treated pigs, not only in vitro. I want to see in vivo. You know, you can talk a lot about in vivo, but go in vitro, go in vivo. And they had a system where they inoculated the drug, 2 milligram per kilogram. You have a 50 kilogram sow, it costs you $1,000. It's not feasible, I understand, but I just give you an example. And then they infect these animals, and they did another one where they treated, let me see, where they did not treat, these were non-treated animals, and then they had contact animals which were treated with the drugs. And what was the outcome? These are the non-treated animals, and these are the treated, directly treated infected animals. That 40% of animals survived, and 60% of the contact animals survived. So there is even in vivo an effect with some drugs which are used against pox viruses. So then people went on, again a Chinese group, to use CRISPR-Cas treatment. And you heard a lot about CRISPR-Cas yesterday, and I just go very fast through the system. You make single guide RNA specific to some regions of the ASF genome. These are the regions in the ASF genome which are targeted. And then you combine it with the Cas enzyme, and then you transfect it into cells, and then you see the outcome. So you have these different sgRNAs, combine it with Cas9, transfected into cells. The cells are infected with African swine fever, and then you see is there an effect or not an effect. And you see these are the different constructs, and there were 3 constructs where they could knock out ASF replication. So now they, again, I like in vivo stuff, they went a step ahead and did the following. They used an efficient construct and made transgenic animals. So they did not knock out anything in the animal, they added the CRISPR-Cas construct to the genome of the animal, and then in blue they infected these animals, and then had a contact animal and 3 wild type. And here in gray or white, they had wild-type animals, infected them with ASF. Here a blue contact and wild-type contacts, and what happens? Nothing. ASF is not simple, right? These are the transgenic animals directly inoculated, no effect. All of them got infected and sick, like the wild-type. There was a transfer from the gene-edited animals to the other gene-edited animals. There was a transfer of the virus as well as from the wild type to the gene-edited animal. So there is no survival advantage of these transgenic ASF transgenic animals compared to wild type. But last year there was an announcement by the Seek Labs from Utah. There is no publication, there is only an announcement from the company, and they say they use CRISPR-Cas-based technology, so similar concepts as I just described in this publication, to treat pigs which were infected with ASF. The study was done in Vietnam with a small number of animals, and they had more than 50% survival. So not making transgenic animals, but going directly into the pig and inoculate the pig. This is the way we follow too in my approach to CRISPR-Cas treatment of ASF. Okay, last part. Gene editing of pigs. A lot is talked about it. We have some successes. The question is obviously, will it accepted by the population eating these animals which are now FDA approved? But time will tell. CRISPR-Cas is a cutting-edge strategy harnessed not only for viral resistance, but also resilience. I believe we will never have an ASF-resistant pig, but maybe an ASF-resilient pig which survives. Because we have to do knockouts. Knockouts are much difficult to do, and you have to really target host genes which are critical for virus replication, or receptors, if you know the receptor. And that's done with PRRS, but we don't know it with ASF. We know it with swine flu, but it's ubiquitous. If you knock it out, the pig is might not survive. So you have to knock out critical host proteins. Or we can use it as therapeutics, which I just described. And it has obviously the potential, and we all know that, to inherently this can be passed down in the germ plasm, in the germline, to make these resilient pigs. So this example you heard, there were two groups, one in my colleague Randy Preeter from Missouri, and the other one, the Roslin Institute in England. They knocked out different parts of the CD163 protein, which is the receptor for PRRS. So if you do that, you get PRRS-resistant pigs. This is a figure from that paper. So this is the wild-type pig in the blood virus, and this is the virus in the knockout. So if you infect these pigs, there's no viral replication with most strains of PRRS. And also no antibody production. The knockout pigs do not produce antibodies versus the wild-type, very nice antibody production. And also clinical signs with the wild-type, no clinical signs with the knockouts. And I started this project 4 or 5 years ago in collaboration with Genus, who actually owns PIC, and to make swine influenza resilient pigs with my student now, postdoc Taeyong, and others. So some is published here in this paper, and one is in revision right now. And what we did was, because the receptor is ubiquitous, it's very difficult to manipulate the receptor for viability of the pig. That's why we decided we concentrate on a protease, a serine protease, which is very important to cleave monobasic cleavage site in the hemagglutinin. The hemagglutinin is produced as a precursor, HA0, and then cleaved by proteases in HA1 and HA2. And we knocked out this TMPRSS2 protease because others before us, we have many smart people. So we had smart people who did that in mice. When they did it in mice, and they used monobasic cleavage site viruses like H1N1, H3N2, H7 and 9, and you have a knockout mouse, they all survive. If you have a wild type, they all die within 6 days. But this doesn't work with H5N1. Because H5N1 has a multi-basic cleavage site and is accessible to furin-like proteases, which are everywhere. This TMPRSS2 is mainly in the respiratory tract and GI tract. So it doesn't work for H5N1, and so we thought, let's study the effect of TMPRSS2 on influenza A replication in pigs. And what did we see? It took us, I became a pig breeder, because you start with heterozygous pigs, and then you need a number of, took several years for me to breed pigs. Not a good one, don't hire me for any pig breeding operations, okay? But I got it done, and at the end I had 9 knockout homozygous and the wild types, and we infected them with 2 important strains of influenza, or subtypes of influenza, H1N1 and H3N2, which are replicating in the field. And when we infect pigs, we always inoculate them intratracheally to get beautiful symmetric distribution into both lung, to the lung, and then we follow them up usually for 5 days, because everything is over after 5 days. But we did longer with other studies. But here we put them down, we euthanize them to see what's happening. I like to see what's happening in the animal at day 3 and day 5, because that's maximum replication in the lung and maximum shedding, and we test shedding, etc. And these are the wild type and knockouts. If you mix them, you will not see a difference. So anyhow, what did we see? What we hoped for. These are the knockout in blue and the wild type in red, and you see after 5 days there's not much virus shedding from the pigs, the knockout pigs, but beautiful replication of both viruses in the wild-type pigs. And similarly, when we look in the nose, there is no virus replication in nasal torpinates in the knockouts versus wild-type do. When we look in the lung, there is replication. So this is not a resistant, this is a resilient, doesn't shed much. You cannot, it's not a resistant pig. So then we did transmissibility studies. So what we did is we had wild-type pigs and knockout pigs, and we infected them. So we did a wild-type to wild-type, but it's our control transmission study. And then we did a knockout to wild-type transmission study. So we wanted to see, is the shedding from the knockout enough to infect contact pigs? What did we see? And we infect them on day 0. After 2 days, we commingle them with the sentinel controls. Then we do some necropsy to see on day 5, do they really get infected? And then we wait for 21 days to look at seroconversion and so on, and we sampled them for this time. So what did we see? This is a typical shedding of a wild-type pig, maximum day 4, 5, 6, and day 8, it's negative. And nothing anymore until there's nothing shedding anymore. If we look at the contact animals to the wild type, you see it takes 4 or 5 days or 3 days before they start to shed, and then a similar replication kinetic. What happens with the knockouts? So knockouts shed a little bit. Do they transmit to wild type? Yes, they do, but with a different kinetic than seen with the wild-type pigs. So there is something there which is different. Then we did a very critical study. What happens if we do knockout-to-knockout transmission? Can we disrupt the transmission cycle of flu in pigs by having knockout-infected pigs, so the virus comes in, infects knockouts, and then you have knockout sentinels, knockout contact animals. This is the critical study. So we infected wild-type and knockout pigs. We had enough at that time. It's not so simple to get sometimes the animals. And then we commingled wild-type with wild-type and knockout with knockout. And again, after 5 days, we look, do they replicate, yes or no? And then we wait for 21 days for seroconversion, and we sampled them over this time. What did we see? We see shedding. Again, this is wild-type shedding. It's negative by day 7 and positive maximum day 4. And similarly, all these, they were transmitted to the 5 sentinel animals, so after a couple days, as we have described before. What happens if we do knockout? We didn't see much shedding of the knockouts, and when we add the sentinels in there, there was nothing. No shedding observed. Now we wondered, is there a problem with our infection? That's why we did antibody tests. So this is shown here. These are the wild-type principal infected animals, nice antibodies after 21 days. These are the sentinel, wild-type sentinel, nice antibodies after 21 days because they all were infected. These are the knockout animals, infected, and you see nice antibody production, so all of them were infected, and these are the knockout controls, no replication. So we concluded from this study that we could, when we knock out TMPRSS2 using CRISPR-Cas, we could reduce shedding. We saw transmission from knockout to wild type, but not from knockout to knockout. No antibodies were found, and we conclude overall that to use genetically modified swine with TMPRSS2, we can minimize the economic losses to the swine industry with swine influenza, and also we can limit the emergence of novel influenza viruses with zoonotic potential in the pig. So we hit 2 birds with 1 stone. It's animal health, you save money in the farm, and human health because it's a zoonotic pathogens. So these are my conclusions. I believe a robust defense system against current and future swine viral threats requires complementary strategies. There is no one-size-fits-all. Not this platform of vaccine works for all viruses. Not this diagnostic approach works for all viruses. We need to integrate not only prevention, but also detection, vaccination, and treatment, and innovation. We have to work together. We have to collaborate together across all sectors. You have to talk to me, and I have to talk to you. That's why I'm here. And only continuous innovation and sustained commitment from the industry, academia, and the federal government, including regulatory agencies, are critical to protect animal and public health ensure food security and maintain the resilience of swine production systems worldwide. So this takes a village. My group at Kansas State, many of them, we worked with the knockout pig with Randall Prater and Chinus and with Nebraska. Pathology, a lot is done at Louisiana State, my 2 centers, and funding is very critical. Without funding, we cannot do the work we do, and some of these studies, especially in high containment with ASF, are very, very expensive. We talk hundreds of thousands of dollars. Without the funding, we couldn't do the work I just presented. Thank you very much for your attention.
Thank you so much, Dr. Richt. If we can put the number again for to submit questions for Dr. Richt, and then we will relay them to up here. We just have just a few minutes for questions before we'll go on to the other keynote. So first question, you alluded that we have made ASF worse by using imperfect vaccines. Is there any evidence that the same may be happening with vaccines for PRRS? And what about as we introduce a gene-edited pig potentially to try to, prevent PRRS transmission?
Okay, so
what might the virus
questions.
do?
Yeah. So first question, yes, the recombinant virus genotype 1, genotype 2, which was first found in China, was most likely created by using illegal vaccination, using a genotype 1 virus. So these viruses, including PRRS, they have the tendency to recombine. And I think even PRRS even more most likely than ASF. So I'm not a PRRS expert, but I can imagine that this happens also in the field with PRRS. I don't know whether vaccines made it worse or better. I cannot comment on that because I'm not a PRRS expert. What was the second question?
About how the virus might respond once we introduce a gene-edited pig population, or if we introduce a pig-edited population.
Yeah, that's a very good question. So if you know, so we looked at, for example, in our system, we look at mutations in the HA always, because that's, if you have a monobasic cleavage site and that changes, our approach will not work anymore. So you have to look for that because viruses are smarter than we are. And the same most likely will be for PRRS. PRRS uses 163 as a receptor, but maybe I'm again not a PRRS expert, they are co-receptors, and then a virus comes up which likes the co-receptor more than the major receptor, and then we are in trouble. So that's biology, right? But we have to think about, these are very good questions. We have to think about how viruses react to different environments, how viruses react to treatment, to vaccines, to knockouts. Viruses have, say, for example, PRRS, how much genome has PRRS? 15?
Yes.
They're so smart with 15 kilo bases, and we have 2 billion bases, right? So, yeah, I had one side digression. I had a mentor, Bill Narayan, and we worked on lentiviruses. He always said, he was a Hindu, I want to be reincarnated as a lentivirus. They're so smart. So I will never forget that. So they're very smart. Sorry for my digressions.
All right, another question about vaccines. So in the US, we have been discussing the possibility of eliminating PEDV. Can you comment on PED vaccines?
Jeez. If somebody here can help me, who is PEDV expert? I know in the beginning they had to feed them dead piglets because there was nothing, right? And now many people work on PEDV vaccine, but that's not my area of expertise. Is there a PEDV expert who can help me?
Okay, fair enough.
Sorry.
All right, what is your opinion on ivermectin antiviral effects?
Oh my golly. Ivermectin, for me as a veterinarian, is an antiparasitic drug. It was very popular during the COVID era to be used as an antiviral, and some people published it works, and others, many published it did not work. So I have not used it. I know my colleagues of mine have used it, even in a human, in monkey, in a non-human primate model, and it did not work very well with SARS-CoV-2. So I don't know, I have never used it, so I cannot comment on it. Sorry. But use it for parasites. That's what I tell you. Use it for parasites. But we have already resistance too. But anyhow.
All right, one final question. Is there anything we can learn from the warthog research in our research for ASF solutions? So what can we learn about from
Warthog
warthogs?
research. Oh, yes. Warthog. Beautiful animals, very dangerous. You saw these heads? Never go in a burrow if you don't dance on top for 5 minutes, because they come out when you jump on their burrows. You land in the hospital. But anyhow, what can we learn? They do not get sick, and they have viremia, especially it's in the young warthogs. So some people, like the Russlin Institute, have tried to make transgenic pigs using some genes from the warthogs, and hopefully making them resistant to ASF. So I think comparative genomics and comparative analysis, why the warthogs doesn't allow replication more than maybe 10 to the 4, whereas the pig 10 to the 4 viruses per mil, versus the pig goes to 10 to the 8, the domestic pig to 10 to the 9, and that's the deadly thing, cannot control it. What are the mechanisms to control it, and why is the warthog, which is millions of years ahead, most likely in evolution to the domestic pig, so efficient? I think comparative genomics and analysis needs to be done. The problem is, to get these critters, you can only get them in the zoos or get the samples into the United States is very difficult, because there is foot and mouth disease, African swine fever, and all this stuff around where these warthogs are living and replicating. They're really like a pest in many of these game farms.
All right, thank you so much, Dr. Richt, for sharing all of your expertise with us.